esterase inhibiting exposures, and we've asked Dr. Alvin Terry to come and talk to us on his recent studies in this field. And he is a tenured Regents professor and chair of the Department of Pharmacology and Toxicology at Georgia Regents University. He also is a licensed pharmacist in Georgia and South Carolina. Dr. Terry received his doctorate in pharmacology from the University of South Carolina in Columbia. To date, he has 146 peer-reviewed research articles, several book chapters, and holds a U.S. patent. His research is supported by many government agencies, including the National Institute of Environmental Health, the National Institute of Drug Abuse, the Department of Defense, and the pharmaceutical industry. Dr. Terry has also served in multiple study sections for a variety of governmental agencies, including the NIH, NASA, the Department of Veterans Affairs, and the Department of Defense. His research interests focus on the role of central acetylcholine or cholinergic pathways in cognition and how these neuronal pathways are involved in memory dysfunction, particularly in relation to exposure to environmental toxins, primarily organophosphates. And that's why we've asked him to talk about his recent studies in that today, finding axonal transport deficits in living animals, and also his prior work in this field. So, welcome, Dr. Terry.
Thank you. Thank you, Dr. Sullivan. It's a pleasure and an honor to be here. This is my third one of these very important meetings. They've always been a very much a pleasure and a learning opportunity for me. So I'm very happy and honored to be able to share with you some of the work we've been doing over the last few years. And toward the end of my talk, I'll focus on a paper we just published earlier this year. But I want to give, as Dr. Sullivan asked me to kind of give you some of the background as well as kind of an overview of some of the work we've been doing leading up to this MRI study I'm going to talk about. And so for most of my career, I guess literally all of it since grad school. Can you hear me okay? Or, okay, that's better. Okay. So for literally all of my career, I've been interested in the central cholinergic pathway in the brain. And if you look at the slide here, this is a rodent diagram, a sagittal looking from the side, diagram in the analogous areas in the human, back up here a second, in the human brain. The cholinergic system is involved in multiple things, autonomic control, but our interest has been in learning and memory and cognition and attention. If you look at the, get my arrow to work here. If you look at this area down here that I'm circling, it's called the basal forebrain. And there are two sort of major circuits in the mammalian brain, this would include the rodent as well as the human, that are well documented to be involved in learning and memory and cognition. This comes from the Alzheimer's literature, it comes from lesion studies in animals, receptor antagonist studies and the like. And the pathways, one would be the so-called projecting from this NBM, which in the rodent is nucleus basalis magnocellularis, a big dollar term, but projects axons to all areas of the cortex and the amygdala. Then this other area highlighted by MS is the medial septum, and they project. There's cholinergic axons to project from that nucleus to the hippocampus and to some other cortical areas. So these are well documented to be involved in cognition, areas we're interested in, both from a environmental toxicology exposure scenario, also degenerative disease, as well as drug discovery. So we're doing work in sort of all of those different areas to both see what diseases and environmental exposures may do that are detrimental to this system, as well as if there are ways to target it from a pro-cognitive standpoint. So another major focus of our lab has been on this class of chemicals called organophosphates, which you've heard brought up by Dr. Steele a few minutes ago, which are well known to affect the cholinergic system. These chemicals, all of them have as a base, or they're all derivatives of either phosphoric, phosphonate, or phosphenic acid. They were first synthesized in the late 1800s by French scientists, later developed as insecticides. by Germany and developed into nerve agents, although there was no evidence they were actually used for that purpose in World War II. But you find this class of chemicals in multiple types of compounds. Most notably would be insecticides, but also, has been mentioned earlier, in chemical warfare agents, ophthalmic agents, anthelminthics, and a number of other products. And so, as far as insecticides, you find them all over the world. They're used in just about every major country to combat insects, and not only in the context of farming and enhancing farming productivity, but also to diminish or decrease vector-borne illnesses. And many would argue that these chemicals have actually saved millions of lives over the years, either by improving farming productivity or decreasing vector-borne illnesses. You know, Western countries like the U.S., Western Europe, you never hear of people dying from malaria or yellow fever anymore. And many would argue that was with the advent of the OP-based insecticides is the reason for this. So that's sort of my good spiel, I guess, about OPs. But we also know that in some ways we've sort of made that old Faustian bargain, or the deal with the devil, because these are very toxic compounds. There's still a lot we don't know about them. And so that's one of the things we're very interested in, is learning more about their toxicology. Now this diagram, if you look at the top, these are how the compounds called organophosphates work, or this is their main mechanism. It's been known for decades that they bind and irreversibly inhibit the enzyme called acetylcholinesterase, which is the main enzyme that breaks down acetylcholine. And so what happens is, if this enzyme is antagonized or inhibited, you have a buildup of acetylcholine in the synapse, and you have too much cholinergic activity. So you have a variety of symptoms that I'll mention in just a moment. I've listed the two major organophosphate classes that we're studying. Bottom left, our representative insecticide that we've been studying is chlorpyrifos, which requires cytochrome enzymes to be converted to its active metabolite, the oxone. And then to the right, we study this chemical called DFP, or diisopropyl fluorophosphate. And it's very similar in structure to sarin, as you can see just above it. And the reason we use that in the laboratory is it's way less toxic, so for laboratory personnel, although it's very similar in structure and activity, just less potent. So whenever you are exposed to a high dose of an organophosphate, this could be a pesticide applicator, a farmer, or someone exposed to a nerve agent. There's sort of this acute high-level exposures. The symptoms are very well documented. The acute muscarinic and nicotinic, these are based on the two types of receptors for acetylcholine. You see the so-called acronym is called dumbbells, but diaphoresis, which is excessive sweating, diarrhea, urination, slows your heart rate down, bronchospasm, emesis, lacrimation, and salivation. If the dose is high enough, you also employ nicotinic receptors, and then you see the muscle fasciculations, seizures, and the like. So we've known about this for decades. This is what happens during acute exposure. What's less understood and what hasn't been studied for as long would be the situation where you've been exposed repeatedly to doses that aren't associated with acute toxic symptoms. And I've used the term sub-threshold here to say, as a sort of an operational definition, would be a dose below that causes acute toxicity. But there have been a number of particularly epidemiologic types of studies that would correlate a variety of behavioral symptoms with this lower level repeated exposure. You can see the list here: anxiety, depression, psychotic symptoms, and then what we're most interested in would be the learning and memory related symptoms. By the way, some of these very symptoms you see in the second bullet here have commonly been described for Gulf War veterans. But one caveat here I would say is that most of this is all correlational. We can't do ethically prospective studies of organophosphates in human beings to see what they do. And so this is the argument for using animal models to make that prospective argument that this one type of chemical, given at lower levels than causes acute toxicity, can have long-term problems. So we know that there are multiple types of possible exposures that Gulf War veterans had. Things, from oil fires to vaccinations to pyridostigmine and prophylactic agents to organophosphates. But if we want to make the causal argument that this alone contributes to the problem, then that's back on my soapbox about the animal model. And that's what we've been doing for a number of years here. The other thing I want to bring out is, so we know the classic sort of canonical mechanism, if you will, the inhibition of cholinesterase. But especially in the context of levels of exposure below that cause acute toxicity, there's quite a bit of evidence that this cholinesterase inhibition mechanism isn't the whole story. And Dr. Lyon at UCSD has made a good argument in one of her reviews, and I won't read all these individual bullets, but to make the argument that the whole story isn't cholinesterase inhibition. There's a lot of different other studies that would suggest that you can't relate all of the symptomatology, particularly with a lower level exposure to cholinesterase. So what are our overall objectives? What we've been doing now for 10 plus years, 10, 12, is looking at what are the consequences of repeated subthreshold exposures to representative organophosphates on these different domains of cognition, and then determine, the consequences of these types of exposures on substrates of cognitive function. So first, can we prospectively make the argument that these lower level OP exposures indeed are leading to these memory-related deficits, and if so, what's the mechanism? If we know the mechanism, obviously we have a much better chance of treating this type of problem. So we've been looking at several different potential mechanisms. They're in our publications, but what I want to concentrate on primarily today will be this third bullet at the bottom there, axonal transport. And of course, our overall goal is to be able to identify therapeutic targets so we can design treatments. Now, this was covered before very eloquently by Dr. Steele, but to make the argument that there is a correlation between organophosphate exposures and Gulf War illness, a number of different papers have been published, one very eloquently by Dr. Gallone, making the argument that exposure to one or more cholinesterase inhibitors is a plausible explanation for a number of the neurological symptoms. This is just reiterating some of the things that were said before. 41,000 military personnel in the Gulf were exposed to high levels of insecticides, and then of course the Comacia incident where 100,000 military personnel may have been exposed. I wasn't actually aware of the uniforms being pre-treated with organophosphates. I thought that was
they're pyrethroids.
Pyrethroids, so it's not OP.
It's not
Okay. So this, I'm sorry. Okay. So this is just a list of organophosphates that were known to have been used in the first Gulf War. As I mentioned, chlorpyrifos is the one that we've been studying for quite a while. All right, so I'm going to go through now, just sort of highlight some different papers we've published. First, to sort of make the argument that subthreshold exposures to OPs do cause attention, learning, and memory deficits that persist, and then go into some of the mechanistic stuff. So one of the tasks that we use quite a lot, I have a core where we do a number of different behavioral tasks, but one we do is a measure of sustained attention. It's called the 5-choice serial reaction time task. It's kind of a mouthful, but it's based on the same theme as the continuous performance task that's used in humans to test for sustained attention. If you look at the bottom part of this slide, you can see that one of the types of the CPT task is called the AX, where if you're sitting in front of a keyboard or have a joystick type thing to punch a button, that the rule will be that every time you see an X that was immediately preceded by an A, you push the button. So obviously you can get a lot of information from that. How accurate are you? Do you prematurely respond? Do you keep responding afterwards? So you get into this thing, how good is your attention? Do you have evidence of impulsivity, evidence of compulsivity, and all this type of stuff? Well, in the rodent, this is a little bit unnatural for a rodent, but it takes about a couple of months to train them to effectively do this task. But if you look on the top left, a rodent is put in an operant chamber and trained to look for a flash of light, and when that flash occurs, to nose poke that spot or that aperture and turn around to the other side and receive a food pellet reward. And so you can measure many of the same types of, same types of dependent measures. Accuracy, which is a indicator of sustained attention. If they omit, that's another sort of evidence of attention or motivational problems. Prematurely respond would be evidence of impulsivity. If they continue to nose poke after the flash of light before they get their food pellet, that's evidence of compulsivity. And a timeout response would mean if they got it wrong and they keep nose poking, then that's sort of this cognitive flexibility, also compulsivity model measure. And you can look at the latency. So for example, one of the reasons why all this is important is if an organophosphate dose you were giving, for example, was just making them kind of sick and not feel good, then they wouldn't be motivated, so they wouldn't nose poke as much, they wouldn't eat their food pellet reward, or their latencies would be affected. So that's why all these different measures are important. But anyway, I just want to show you a couple of slides from a paper we published in 2010, and this is where we took the organophosphate chlorpyrifos and administered it every other day to rodents at a sub-threshold dose. I think this was at 18 milligrams per kilogram. And if you look at the slide, you can see these animals are trained to where they're around 85% correct. And you can see the open boxes there are vehicle controls, and they sustain their level of accuracy throughout the 30 days of vehicle exposure. But you see the animals exposed to chlorpyrifos, their accuracy goes down, and it's significant across the whole 30 days of the rest of the 30 days. And then we wash the animals out. In other words, we stop giving the chlorpyrifos and we look at recovery. And this is another 30 days after that. And you can see that they never fully recover, although they do abate, or the performance doesn't improve somewhat. So this is evidence of sustained attentional deficits that are persistent. The other notable finding for this particular study was that these animals prematurely responded. And if you look across the first 30 days, you start to see increases in premature responding, meaning they can't wait to the flash of light and they're nose poking. And then that persists throughout the rest of that 30-day washout, meaning it's impulsive-like behavior, which is certainly a behavioral finding that hasn't been shown before this study in animals. Now, so the conclusions here that repeated exposure to low-level chlorpyrifos results in impairments in sustained attention and increased impulsivity. I will mention that word low-level kind of got us in a bit of a argument with Dow Chemical. They didn't like the concept of low-level, but I should have used that term sub-threshold here. But so I would argue that yes, that wasn't really low-level. It certainly wasn't trace levels, but it was below the level of acute toxicity. And then more recently, we did another very similar study with a sub-threshold dose of this DFP, which is a nerve agent type. Similar findings. I would say that these animals did recover, and over the course of that 30-day, they did come back pretty close to normal. And one argument would be here possibly, and we put this in the paper, that these lipophilic insecticide type OPs like chlorpyrifos tend to linger longer in the brain. We've shown that, especially the ones that have a chloride component. And it may be that the nerve agent at this lower level, it may be that its activity doesn't last as long. But I'll show you a paper in a few minutes where we argue against ourselves on that same argument. And then here, the other notable finding was we saw this big increase in timeout responses. We did see emissions and some other things, but the other notable finding was that these animals would, if they got the problem incorrect, they would continue responding through this timeout, which is that cognitive flexibility, compulsivity type of component. So similar types of outcomes And so then the conclusion of this was that repeated exposures to DFP result in impairments of sustained attention and persistent alterations in inhibitory response control. All right, I'll show you another paper, and I think this may have more punch to it, if you will, just because of the time, the amount of time we washed out the animals and we're still able to detect impairments. And you've probably seen similar slides like this before, but we do one of the classic hippocampal-based spatial learning tasks of reference memory called the water maze, or the Morris water maze. And this is where you take an animal and place it into a naive animal that hasn't been placed in a pool like this. It's a big open field pool of water that has a hidden platform in it. The water can be either opaque, or you can have colors like black on black so the animal can't see the platform. They swim around for a while, and finally will stumble onto the platform, look around in the room and see spatial cues and so forth. Come back with another trial and you keep repeating it. Eventually they get very good at it. You see on the right side where they're pretty much going straight to it. And this is a classic water maze task. So here are data where we looked at chlorpyrifos, a couple of different subthreshold doses, again every other day for 30-day exposure. Then we gave them this very long washout, which is 140 days. And you can look at the left side of the slide here. Error to show up here, but you can see that a normal control animal, that they quickly learn over time and develop, their latency to find the platform decreases. They get down to 10 seconds or so by 5 days of 2 trials a day. Some impairment with the 2 doses of chlorpyrifos. Then we did a second, if you look at the top left here, one of the things you have to measure is swim speeds, because if for example, the drug or organophosphate was causing some kind of motor deficit that would contaminate this type of task that's reliant on swimming behavior. You see there's no change in swim speeds. The right side of this figure, we switched the location of the hidden platform, and we do a relearning phase, and that's to kind of get at this cognitive flexibility. Well, you see here that the chlorpyrifos animals did not have any problem with that second part of the study. Conversely, when we looked at DFP, same paradigm, same 30-day, every other day exposure to subthreshold doses, pretty robust impairments with DFP during that exposure period, or after the exposure period. And then on the right side, you can see that they actually did have deficits at learning the location of the new platform. So this gives you this idea that their flexibility of thought, or their ability to learn when the rules change, they're not picking up this new rule and taking it forward. So there's sort of cognitive flexibility deficits. And again, this was 140 days after their last organophosphate exposure. And this is just to show that with the higher doses that the cholinesterase had fully abated by 45 days. So it's clearly not associated with cholinesterase inhibition being occurring while we're doing the behavioral testing. So the conclusion of that study was that repeated subthreshold doses to chlorpyrifos and DFP may lead to chronic deficits in spatial learning and memory long after cholinesterase inhibition has abated, and that insecticide and nerve agent OPs may have differential effects depending on the domain of cognition. So maybe all OPs are not alike. All right, what about this business about axonal transport? So there's been quite a, I guess, evidence over the years to argue that this very fundamental component of neuronal function can be affected by OPs. There were some papers, I think it was in the 1990s, maybe in late 80s, by a group at Duke, Abodonia, and colleagues that did this optic nerve preparation where they gave the rats very high doses of this highly neurotoxic OP called THCP, and there was another one where they saw that growth factor, axonal transport of nerve growth factor was decreased. And that paper just kind of laid around. You didn't hear much more about that for a long time. But and later we were doing studies of OP exposure and finding that cholinergic receptor expression could be altered by the same kind of paradigm with organophosphate. But in particular, the receptors that are known to be on the presynaptic side tend to be diminished, and these are like nicotinic receptors and M2 muscarinic receptors. You got to think, well, maybe there's, you're not getting out to the bouton. And so we did a series of experiments at that time in rats where we would inject them with similar doses of chlorpyrifos that I showed you, and then do washouts and then take out their sciatic nerve, and under a DIC microscope you can watch vesicles moving across a window on the screen and have a blinded observer just count the vesicles moving across the screen. It's kind of a neat technique. We collaborated with a guy named Dale Sickles, who was a big acrylamide expert at the time looking at axonal transport, and we started. So we found clearly that these vesicles being transported both in the anterograde and retrograde direction were being inhibited, and that this lasted up to 14 days after the last dose. But that's a peripheral sciatic nerve. That's not anything to do with the brain, or not anything to do with learning and memory. So eventually we wanted to get into memory circuitry. But other evidence that we published that would suggest that transport was affected and that what might be a mechanism. We and others have published that tubulin polymerization. I'll say a little bit more about that in a few minutes. It's very important for formation of microtubules, which are important for axonal transport. That tubulin polymerization was affected by OPs, and that Dr. Oksana Lockridge and others have done a lot of work looking at other kinds of molecules besides cholinesterase being covalently modified by OPs. She has a whole list of those. I'm now collaborating with a chemist up in Athens, Georgia, who's also working in the proteomics area to look at these covalent modifications, things that would affect the machinery that's involved in axonal transport. And this is a big ugly picture of a neuron, but I've sort of stretched it out so we could talk about some of these organelles. So you hear me say a lot about axonal transport. What's the big deal? Well, it's a fundamental process in neurons, and so whenever something is, a protein, for example, is made in the endoplasmic reticulum, is packaged by Golgi into vesicles, and the kinds of proteins we're talking about could be neurotransmitter receptors, they could be neurotransmitter transporters, synthetic enzymes, catalytic enzymes. They have to have a way to get out to where they need to be, so out to the terminal bouton out here to the far right. Also, mitochondria, which are the energy-producing machinery in the neuron, has to get to its site where it needs to be to support a variety of different chemical processes that require energy. So both the vesicles and mitochondria have to have a mechanism of moving out axons and getting to where they need to be. So there's sort of this railroad track, if you will, called the microtubular system that's responsible for the transport. So there is a type of motor protein called kinesin that's best characterized at moving these vesicles and mitochondria in the anterograde direction going this way, and then dynein is the motor protein responsible for moving in the retrograde direction. For retrograde transport, it's very important for growth factors to be transported from their target. They're taken up by the bouton and transported retrogradely back to the nucleus so they can have a trophic effect. So all this is critical to the functioning of a living neuron. So what if something is affecting that? We would argue that would be a fundamental problem. And this is just another diagram taken out of a review article I put together a couple of years ago, showing you this is the diagram of the microtubule system. These microtubules are made up of dimers of alpha and beta tubulin that then polymerize to form this, what we call a railroad track. Then this is a kinesin motor protein taking its cargo, and conversely, or in addition to that, dynein moving in the retrograde. So our argument or our hypothesis is that these very reactive molecules called organophosphates can either bind and covalently bind kinesin and affect its activity, or they can affect this tubulin polymerization process as it's highly responsible or involved in formation of the microtubule. And the same kind of concept would be down at the bottom, possibly with dynein, although we have not studied that. All right, so the first thing we did to get into brain neurons as opposed to sciatic nerve was we did a study in cultured neurons where we monitor the movements of mitochondria. And I mentioned to you before, mitochondria have to be axonally transported. One of the notable things here, and so we use this fluorescent label called MitoTracker that labels mitochondria very nicely in these neurons. But one of the first things when Louise was a graduate student at the time was doing the study, she came running in my office and showed me a slide, this video, and said, look at these mitochondria and see how elongated they are. And when I was in grad school or in college, we always taught mitochondria is a sort of bean-shaped organelle that's really active as the energy machinery. I didn't know about all this elongation, fission, fusion, and all this type of stuff. So this wasn't my area of expertise. But anyway, you see this robust change in their elongation. Essentially, they were not breaking apart, or they were fusing. And so that was one interesting finding. But nevertheless, she was able to track their movement. So under a microscope, you essentially form or you pick a region of interest, usually it's just out from the axon hillock, and you monitor their movements, which we defined as number of moving mitochondria per micron. The criteria would be they had to be moving in the same direction, and so in other words, and both ends had to be going in the same direction, because these things can kind of quiver. We also looked at the length and the number of mitochondria, and this is just an overview of our take home from that paper. Essentially, we found that the movement was decreased, their length increased, and their number decreased, both with the chlorpyrifos chemical as well as its active metabolite. When I say active, what's known to inhibit cholinesterase. But you can see on the bottom that the doses or concentrations are much lower where that threshold is. So this essentially says that the mitochondrial morphology is being changed as well as their transport. We also did several studies to see whether or not there was maybe just some overall more or less specific effect on the neuron or the mitochondria rather than just this fission fusion. So we did the so-called decipher assay, and this is essentially it's a type of chemical. It's our lipophilic cationic molecule that in its monomeric state is taken up into a neuron and fluoresces green. So you can see at the top here. But when it gets into the neuron and comes in contact with normal functioning mitochondria that have a normal membrane potential, then it'll begin to dimerize, polymerize, and turn to where it fluoresces red. So you can see that under normal conditions you'll have a nice green and red fluorescence. And you just do this ratio of red to green just to essentially get an idea of how functional the mitochondrion is. See, out to at least 20 micromolar, we didn't see any change. This was the highest doses we tested in culture. If you go out to we had to go out to 500 micromolar to actually see where the red fluorescence was decreasing. So the argument here, when you can see the blebbing and apoptotic sort of manifestation at the very high concentration. So the argument here was that our effects on mitochondria were not related to just non-specific damage to mitochondria or non-specific problems with the neuron. We also did ATP studies and we did superoxide studies, and at the doses we used we didn't see any changes in those, indicating that our effects on mitochondria were specific. So the summary here was that we did see concentration-dependent decreases in transport of mitochondria, and then increases in their length and decrease in number, which is indicative of increased fusion versus fission. I didn't show the data, but these changes did occur at concentrations below that impair cholinesterase. It wasn't related to ATP problems, mitochondrial membrane potential problems, or superoxide production. And so the take-home was that an underlying mechanism of organophosphate-based neurologic functional problems might involve mitochondrial dynamics or their transport. Now I'll just say that I'm not a mitochondrial expert. We are collaborating with some other people to follow up on this fission dynamic thing, but that's not my thing. So we wanted to get to a more specific model. And so the next few slides I want to show you is where we've put together a new model with the help of a collaborator up at Cornell named Jordy McGrain, who has created this version of the amyloid precursor protein. You've probably heard of that in the context of Alzheimer's disease. But it is a protein that's well documented to be fast axonally transported in neurons. It's used a lot in studying AD-like models. But we were able to get this construct from our friend at Cornell, where the APP is connected to a fluorescent molecule called dendra, which shows up in coral that fluoresce. And there are different ways you can do the fluorescent label, but he has his construct. We got it from him. You can see at the bottom, this is just a histologic slide showing, you look at the arrow, showing that you nicely label these vesicles that contain this fluorescent protein. Then we use time-lapse imaging to actually monitor their movement of these vesicles over time. And you take these, make these videos, you import them into a program called ImageJ, and it has a feature where you can construct these chymographs. Essentially, it stacks up the images and then gives you can have this sort of more or less time-distance relationship. It's hard for me to use this pointer here, but if you look at the bottom, so essentially what you do is you see the vesicles going in this direction, going from top to bottom, then that's antegrade, and you draw these lines that gives you the distance they've traveled over time. Retrograde is the other direction. So you can measure very accurately the microns traveled per unit time. And when we first set up this model, we used a positive control called colchicine. If you've ever had gout or known someone that had, this is a drug that's been used for that for many years. It's been around for thousands of years, but it's known to impair tubulin polymerization, so it's commonly used as a positive control in these axonal transport studies. And this is just a dose effect or concentration effect curve. You can see that the velocity of these vesicles were impaired in a concentration-dependent manner, both in the antegrade and retrograde direction. So that was sort of validating our model that we could actually measure axonal transport and its impairment by a positive control. And this is just a video to show you this. Control. If you look at the top right there, you can see the vesicles moving along. And then this is them being slowed down with 10 nanomolar colchicine. If you can look back and forth, you can see the difference in velocity. And so the next series of experiments was to look at this nerve agent called DFP with 1 hour exposure. And again, you see a very similar profile of a concentration-dependent decrease in both antegrade and retrograde axonal transport. And you can see that our threshold for impairment is around 0.1 nanomolar, so that's a very, very sensitive effect, well below the threshold for cholinesterase inhibition. This is a video of vehicle again, vesicles moving, and in DFP, if you spot those little vesicles moving along, you can see they're moving slower. That's the sort of take home with that. So to make the argument that this was not somehow related to just cholinesterase or cholinergic effect, and oh, by the way, we also did 24-hour exposure. You got the very, very same type effect. And that's shown in the top figure there, both antegrade and retrograde. So we took a concentration that was reproducible, the lowest concentration or a very good concentration that was reproducible in impairing transport. And then we incubated it in the presence of both a muscarinic and a nicotinic acetylcholine receptor antagonist. And if you look at the bottom there, essentially can tell that the drug mecamylamine or the other drug atropine did not affect this impairment of axonal transport, saying this is a receptor, a cholinergic receptor mediated. By the way, if you have any questions, don't hesitate to interrupt me. I'm kind of moving along.
Yes?
In the work that I've heard presented by Dr. Morris, Marion Morris, and by Jim O'Callahan, to make this model go from an acute model to a chronic illness model. One had to include a stress hormone or a stress event at the time of the exposure. When you're exposing these animals and measuring these things, are you exposing them and measuring them shortly after the exposure, or are you letting some months go by?
Well, in the animal, we have the low-level exposure, then we do behavioral testing after washout. I mean, almost all of it we've done is after a long washout. That's what you're...
Sort of, just curious if you've ever used court as a...
No, we haven't. Plenty of other people have. I mean, corticosteroids are increased by OPs. I think Pamela Lyon and lots of other people have done that. We don't do that in our studies. So, I mean, you could, in the culture model, you could probably just put corticosterone in there, but we want to keep this as clean as we can there.
It's just, to me, it seems like when you're talking about transport, it's something the court would affect. So,
Yeah.
You've proven now it's not a receptor, and it's not
Yeah.
specifically...
That's a good point. I don't know whether corticosterone, that's what you'd use for a rodent, instead of cortisol, you'd use corticosterone. I haven't seen axonal transport studies with corticosterone, but it's a good possibility, good point. Yeah, okay. We also, in this set of studies, looked at cell viability, and we did the classic so-called MTT assay, this is a spectrophotometric assay on the top. You can see up to 100 nanomolar DFP. We didn't see any change in viability. The slides to the right essentially show you these different stains we do to look at cytoskeletal integrity, somatodendritic integrity, and so forth. And basically, you can see from left to right, vehicle to DFP, there's really no change in the morphology of the neuron. The cytoskeleton isn't disrupted, and so forth. That this isn't just some type of overtly toxic effect on the neuron. So the conclusions, these are unpublished, by the way. We hope to get this out maybe in the next month. That DFP impairments in axonal transport in these series of studies were concentration dependent. They occurred at concentrations that did not inhibit cholinesterase, were not blocked by cholinergic receptor antagonists, and were not associated with compromises in cell viability. All right. Now to the punchline of the day, I guess, is our MRI studies. And so we have now set up a technique that involves my laboratory as well as our small animal imaging core at my university, where they do small animal MRI and other imaging techniques. And we've adopted a technique called manganese-enhanced magnetic resonance imaging. So manganese is a very nice contrast agent. It's a divalent cation like calcium. It's known to be taken up into neurons through voltage-gated calcium channels, packaged into vesicles, and people have shown that it is transported by fast axonal transport. Because of its paramagnetic properties, it makes a nice contrast on T1-weighted MRIs. And the pathway that we've begun with is this, so if you look on these are horizontal images, so looking from the top down, and you see the arrow pointing to the optic nerve, that what we're basically exploiting the well-known visual pathway where the retina, which is in the eye, is connected to the optic nerve, and it goes, if you follow it, get my arrow going here, passes through what's called the optic chiasma, and eventually goes back to an area of the midbrain called the superior colliculus. And you can see the optic nerve on the injected side. And I'll show you this technique in just a minute where we do the injection. Nicely labeled, and then the superior colliculus on the opposite side, contrasted. And this is the technique. So you essentially take a solution of manganese chloride, take an animal that's anesthetized with a very thin needle, inject this manganese solution into the eye. You can see that if you follow the red line there, that crosses over to the other side of the brain, and its target or the lateral geniculate in the thalamus and the superior colliculus in the midbrain. So it's a very well documented circuit, and it's been shown that this manganese tracer very nicely gets to the superior colliculus in about 24 hours. And you can measure the optic nerve typically around for up to about 6 hours. It's been used in the aging literature, the Alzheimer's literature, and frontotemporal dementia animal models to show that transport deficits are decreased. So we adopted it to look at what organophosphates might do in this particular model. And again, we used our positive control colchicine, which I talked about briefly earlier. If you look at the top figure, you can see the site where we injected the manganese, nicely labeled optic nerve. And you look at the bottom on the left, you can see that it gets out just for a few millimeters, but that's as far as it goes. So it really locks it down. It only took 3 animals to show a highly significant effect. Again, this is a molecule that inhibits tubulin polymerization. It's a positive control for impairing axonal transport. The data to the right, or where you actually do this, is similar to densitometry measurements, but there are a lot of pretty complicated formulas to normalize the intensity, where you use, there's a whole lot of different, use the contralateral side and other areas like the cerebellum to use as background subtractions. But at any rate, you can see the open circles there with vehicle that the intensity is higher throughout the whole length of the optic nerve to the optic chiasma, whereas with colchicine is way lower. So take home is it's radically impaired. And you look back at the superior colliculus, the top view there is in the sagittal plane, so that's looking in this direction. And then the bottom images there are horizontal, or what we call axial. You can see the nice labeling of the superior colliculus in the vehicle side, and then you see it's mostly absent in the right side with the animals. This is actually an intravitreal injection of colchicine, so we shot it in the eye at the same time we put the manganese in there. Colchicine is really toxic, so we try to do that peripherally. We had a couple animals die, so we quit doing it. Just went with the positive, go straight into the eye. Again, on the right, you see, then this is at 24 hours when you look at the superior colliculus. So we started the chlorpyrifos studies with a subthreshold dose of chlorpyrifos given acutely. So here you have the animal anesthetized, and you do the eye injection of the manganese and the chlorpyrifos injection subcutaneously peripherally at the same time. And take home was that we did not see any impairment. This is a bit of a surprise because back when I mentioned those sciatic nerve studies we did a few years ago, we did see a decrease in the sciatic nerve after a single injection about 10 hours later. It says something's different about the sciatic nerve, maybe in peripheral injection, the chlorpyrifos can get there quicker or what have you, or there's something different between optic nerve axons and sciatic nerve axons. But anyway, that was a bit of a surprise. And again, at the 24-hour time, there was no change at the superior colliculus. The next series of experiments we did was a 14-day repeated exposure to chlorpyrifos series of experiments. Again, so here we did baseline measurements. We did the 14 days of exposure, then 24 hours later we did imaging again, and then 30 days later we did imaging. This was a bit tricky, but we did the full analysis. If you can see over to the right, kind of surprising, but we actually had statistical impairments. Get back up here again. At the 3 milligram per kilogram dose, up to about 4 millimeters out from the eyeball. In the higher dose, it actually did not come out statistically significant. So there was a little bit of a head scratcher as to why this might be, although you can see at the bottom all of the dark circles are below control, but they don't come out significant. There may be some issue with the 6-hour time point. I mean, one of the things that has been published that the rate of manganese transport in the optic nerve is about 1 millimeter per hour. So it may be that there's just too much labeling. If you see you're going out to 10 millimeters here, maybe there's just too much labeling, and then maybe we need to do a shorter time point. However, at the 24-hour time point, we clearly saw deficits. And if you look at the top, so we do and each animal has a baseline, then it has 14 days of chlorpyrifos exposure, and 24 hours later we image, and then we image the animal again after a 30-day CPF-free washout. And if you look at the most notable slide there is the one at the bottom, which is the 18 milligram per kilogram dose, where we saw impairments both at the first washout as well as at the 30-day washout. And keep in mind that that doesn't look like a big difference in number, but that 1.4 it would be about 40% increase in contrast. So if you're looking at the the number down to just over 1.1, that's actually percentage-wise, it's a big change. And you can tell, you can barely see any contrast in the animals to the right at the bottom in the superior colliculus. All right, so conclusions of this study. There we did a lot of other stuff in there, but there's the intravitreal injection of this positive control led to marked impairments of axonal transport, sort of validating that we could detect this in our lab conditions. The single subcutaneous injection of chlorpyrifos did not appear to affect the transport, but then conversely, the repeated subthreshold exposures led to prolonged impairment, so up to 30 days after the last injection in the living rodent brain. So these are live animals. The overall summary of what I've shown you so far from all of our work is that repeated subthreshold exposures to both insecticide and nerve agent OPs lead to protracted impairments in memory-related tasks, and we've done a lot of this. So both drugs in water mazes and radial arm mazes and attention-based tasks. So we now have, I think, pretty credible evidence that these compounds do lead to prolonged memory-related impairments at doses that don't cause acute toxicity to begin with. That there's some evidence that they might actually have differential effects depending on the domain of cognition. This we found to be kind of interesting. We don't know, it could be just something as simple as the fact that nerve agent type OPs are so highly reactive they kind of do their thing and then they go away, whereas the pesticide types tend to be more lipophilic and hang around longer, and so they may leach out of fat over time. So there's a lot of different hypotheses why this might be. And then one non-cholinesterase mechanism may be that these compounds are impairing axonal transport. Say just a little bit about, I'm not doing on time, but we're okay with time? Okay. So just say a few words about some of the other things we have going on in some of our future studies. By the way, so we also, in parallel with all this going on, we're working with this guy, Michael Bartlett, up at the University of Georgia. And right now we're taking purified proteins like tubulin that you just buy and basically expose them to trypsin to break them up, and then we incubate that with chlorpyrifos oxone to see whether or not there are sites where the organophosphate can stick on this tubulin. Remember, that's very important. The tubulin has to be polymerized. The alpha and beta monomers have to be polymerized to make the microtubule. And so one hypothesis is that these OPs are sticking on to parts of these tubulin molecules. And what's reflected in this particular mass spectrum here, this 274, is actually a fragment where this component of beta tubulin is actually covalently bound with the chlorpyrifos oxone. So there is evidence, at least in that very artificial in vitro environment, it's clear that OPs can stick on other things besides cholinesterase. One of the other things, remember when I showed you the very first slide, I talked about the importance of this so-called septohippocampal system for learning and memory. So we've been doing this optic nerve circuit, which is just easier to access with our magnetic resonance imaging study. So, and also in the in vitro models, we've been using cortical neurons, but we wanted to go to the septal neurons, which is they're so well documented in learning and memory. So we're culturing septal neurons now. This is just an example of, I think this really shows you those vesicles moving along. I think that's pretty cool. But anyway, this is our where we're headed with a culture, which would be more relevant to our memory circuitry. That's why I'm saying that. So the work I showed you with all the magnetic resonance imaging is a CDMRP funded project. The AIM-1, I've just shown you half of AIM-1, which is chlorpyrifos. We also have DFP, several doses, and we're just about to break the codes on all that, and hopefully we can publish a DFP paper relatively soon. Don't know what the results are yet. The other part of that study, which we also have ongoing, is we're doing 2 measures for myelin. One is a type of MRI called diffusion tensor imaging. Those studies are also ongoing. And we're doing black gold histology, which is in the animal that's sacrificed, but we're looking at, we can really go in and look at individual fibers and see whether or not their myelin is affected. And this is just one histologic slide. I shouldn't even say much about this because this is, again, is blinded. We're doing unbiased stereology, so this is very time consuming. But just looking at that stain, you can kind of look to the right in the areas of the hippocampus, and they see those individual fibers seem to be labeled with less of this black gold stain, which suggests there is myelin being affected, but I'll hesitate to say more until we break all the codes and everything. We have a number of other things we're looking at. I won't say too much about these, but other possible targets for organophosphates. Tubulin acetylation is something that's well known to promote axonal transport, and it might actually be a target. This is being looked at in the Alzheimer's and the Huntington's disease field. There are compounds out there that are called HDAC inhibitors that can enhance tubulin acetylation, so that might be a way to improve this axonal transport problem. There are microtubule stabilizers. There's one compound, there's a lot of publications out now. I'm not sure I'm pronouncing it correctly. It's called davunotide. It's known to stabilize microtubules, very much been shown in a number of Alzheimer's related models. We think this might be relevant to this OP exposure axonal transport problems. That would be a possible mechanism of treating OP related axonal transport deficits. We're looking at other things, signaling proteins and so forth. We also are developing another type of operant task in our lab for behavior. It's a type of for executive function, which is more of this cognitive flexibility, but a lot more detailed way called attentional set shifting. We're also looking at going to actually injecting a manganese tracer directly into the medial septum. So again, to get away from just the optic nerve circuitry, which is not really traditionally thought to be learning and memory related, to this defined circuit we know is definitely. And our preliminary data shows that we can do it. If you see on the left, that's the site of the injection with the manganese tracer. Then on the right, the hippocampus on both sides is nicely contrasted. So we believe we can do this. It may take a while to work out the details, but that's what we have in future proposals to look at, which would be to do any MRI with the septohippocampal pathway. Okay, just to acknowledge folks that actually did the work that I showed you. This is my pharmacology lab, directed by Katerina Hernandez. Gia Gao did all of the in vitro axonal transport stuff I showed you. The rest of the folks worked on a lot of the other parts of the project. This is the group that does the small animal behavior. And then finally, this is not relevant to this, but I also have a non-human primate behavior lab. We don't do any organophosphate studies in monkeys. It's all drug development, pro-cognitive, but just to acknowledge those folks. This is a person that keeps me from losing my sanity, my administrative assistant. And I mentioned Dr. Bartlett, who's my analytical chemistry colleague. Nathan Yanasak is the medical physicist who's been assisting us with all the MRI work. He runs the core for small animal imaging. Jordy McGrain gave us the APP dendra construct. Other folks that have contributed, previous lab members, and then my work over the last few years has been funded by NIEHS and the DOD, particularly the CDMRP. And I appreciate your attention. Thank you.
Thank you, Alvin, for that great presentation. And just to put some of your work in context, I think, shortly after go forward, and it was quote-unquote common knowledge that if you didn't have an acute cholinergic crisis and poisoning from these types of exposures, pesticides and/or inert gases, that there were no chronic effects. And I think your work is systematically shown in cells and behavioral studies with animals, and now actually tracing in the live animal that is not true. And that's really a phenomenal work to actually show this systematically in the most relevant exposures the Gulf of Veterans had, chlorpyrifos, DFP, sarin, surrogate. I think that's really tremendous work, and it just is pushing forward and forward each stage to look at, what exactly is happening. And this is not something we're able to do in the veterans themselves. We can't trace for axonal transport, we can't really image it that way, so this is a case where using the animal studies is incredibly important. I think one of the things you've also shown is that you take away the exposure and then 30 days later, and sometimes even longer, you're still seeing effects. Can you remind us 30 days in an animal, how long is that in a person?
Well, typical lifespan for rodents is about 3 years. So I always know this because we do antipsychotic studies and we do them for 6 months, and that's a fifth of the rodent lifespan. So I guess you're talking about still a matter of years.
Years, exactly,
Yeah,
right.
Multiple
So when people,
years.
I just want to make it clear, when people hear 30 days, oh well, 30 days, maybe,
Yeah.
it'd be gone.
So in a
It's supposed to
rodent,
be
that
for a long
represents a
time.
much higher.
This is many, many years if we're talking about the human analogy to that. So I think this is, just to put it in perspective, how important this work is. It's really, really important.
Thank you for those kind words.
And I think the idea of then saying, okay, what do we do about that? What do we do for treatments? I think your idea of thinking of microtubule stabilizers, NAP, is really important. Well, that's exactly right. So Dr. Taylor was saying there's some ideas, there's some things that stabilize the microtubules, if that's the problem. There are other things that I think are worth now starting to look at. Do you have plans to do that in some of your new proposals?
We do in the culture environment to start with, and so we have to do a lot of concentration effect. So yes, we want to do compounds like delphinitide, but there's some Taxol, which are sort of related to chemotherapeutic drugs, but that also stabilize microtubules. So we'll start that in culture, which is a long ways obviously from human, but we want to make sure that it works. That we don't. The worst thing you can do is go through and throw something out in clinical trials that hasn't been vetted properly. So yes, the answer is yes. We definitely have plans to test these different treatments. There are a few other things that I guess we also should add here, and I've been asked this question a lot, is this axonal transport effect specific for organophosphate inhibitors, or drugs normally thought of as cholinesterase inhibitors? So we also have studies ongoing with reversible compounds like donepezil, which is used for Alzheimer's. We didn't propose to do pyridostigmine, but that's probably something we ought to do to see whether it's really, so if that covalent modification we're talking about with OPs really is the whole story, whether it could be some effect that's related to how these compounds affect cholinesterase. So that's one other.
You plan to do a combination of exposures?
Not yet.
Not yet.
No, that would be further down the road for us, I think.
And of course,
What we want
there's
to
other
do.
OPs we've talked about too that.
Yeah, and different
just
OPs. I
looking
mean,
at,
not
yeah.
just chlorpyrifos. I mean, we want to look at other insecticide type compounds. The ones that don't have that chlorine would be, because that's one of the big arguments about persistence. Is the ones that are halogenated type may be more liquid soluble and hang around longer. What about non-chlorinated pesticide OPs? But the combination stuff, I think, will be a while for us. We want to publish all the DFP stuff to see if there's a difference in the types, and then move to that kind of thing.
I think your point of all OPs are not alike is, I think you're right. I think we've seen that in our, in our pesticide applicator study, that we found that combination exposures were bad for behavior, but yet individual exposures to particular pesticides also had a distinct effect as well. So that was really important for us to learn that we didn't necessarily know that going in, that would be the case. So I think you're showing exactly the same thing in your animal model, which is really, really interesting. And it will help us to then say, how do you do treatments? Do you have to be selective based on exposures or combinations? And perhaps we will be. So, very nice. Thank you.
Was there a, Beatrice, was that, did you have a
I just
question?
want to ask, there are some studies actually in fish, and I don't know if that makes a big difference, or I think it was maybe an eel, showing that it can protect against acute lethality and also chronic effects of organophosphates with with exposure to good quality antioxidants either just before or just after exposure. And this is also a non-cholinergic mechanism. You mentioned that you didn't see any relationship to mitochondrial superoxide dismutase release, but have you identified any other mechanisms that might account for oxidative stress as a major mechanism of toxicity?
That's a very good question. We haven't, but I know there are a lot of other people that are looking at the oxidative free radical story. And certainly the types of tests we did in that mitochondrial were just kind of, I shouldn't say crude, but they're the standard sort of off-the-shelf assays. You know, there's a lot more to mitochondrial function than the kind of, and that's we're actually collaborating with a guy named Bobby Thomas now, who's more of a Parkinson's guy, but to get into all the different dynamic, you have these dynamic-related proteins that are involved in the fusion-fission dynamic. You've got all the other things they do that's related to oxidation. We haven't pursued much of that, but it's an excellent question, and in fact, people have challenged us, and maybe this is all just oxidative free radical related. And another thing we probably should do is bubble in some low amounts of hydrogen peroxide or something to see whether you get a big axonal transport change. I'm sure you would if you blast them, but you'd have to do it at a very low level. But certainly that's a good point.
Really nice, really a little bit more. I was interested in your mass spec ex vivo, well, in vitro, I guess. I wonder if you're going to do, are you planning to do any ex vivo work, like doing pull down from the brains and then seeing if they've had any effect on PPMs?
Absolutely. It's proposed in our renewal for NIHS. So, yeah, absolutely, we want to go there. And the other thing that this is very difficult to do, but we really would like to, and we may be able to do this with a separate hippocampal approach where you only inject manganese one time, is to be able to correlate in the same animal a behavioral deficit and the transport. With these multiple, when you're doing these multiple injections and multiple anesthesias, it's just not really practical to do it with this optic nerve thing we've been doing. But that would, I think, both would make our argument better, certainly, if we could do that in the same animal.
Other questions? So mine has to do with the pathology here. You showed maybe some white matter pathology, which could be interesting in terms of a free radical mechanism, perhaps. But can you look at APP?
I can't hear.
Sorry. I'm not an expert in how one looks pathologically at tissue following a tissue that has a primary disorder of axonal transport. Some people have used aggregated APP staining. So I thought that that's where you were headed, but you were using APP as a visualization marker.
Yes.
But you can actually stain aggregated APP as a marker of axonal transport. There may be many other ways to do it, but I'm just thinking about a pathology that one could then deploy into human tissue to see if one could find a similar axonal transport pathology.
Yeah. Well, we've actually done those kinds of. I actually have a slide I didn't put in here, but we've done the precursor protein immunostain in rats, and so what it shows is in a normal rat, when you stain for APP, you don't see very much unless there's something holding up its transport and it becomes immunopositive. So we actually have an unpublished study in the hippocampus of rats given DFP where you can show it, and that's an indirect measure. Now, getting into the patho, I don't, in a human brain would be, people make arguments about axonal transport in AD all the time. A lot of it is based on amyloid transgenic mice and just doing similar stuff to this. Whether you can actually do some kind of stain that would, because an Alzheimer's brain is so loaded with 80 plaque. I mean, I've never seen anything of if there were any post-mortem studies and veterans that passed away. I haven't seen anything like that, but.
So in other diseases, people have made the claims in multiple sclerosis, for example, you could see APP aggregates, and you can even see it in formalin fixed paraffin-embedded tissue.
Yeah, and this is the precursor protein, not the A-beta-142 neurotoxic fragment, APP. It's very interesting. I think if you did the same type of stain that we did for the RAC, the APP stain, I'm sure it's been done, but I don't know the answer in the Alzheimer's context. But that was definitely a good point to do that. But to answer your question, I think I may have that slide somewhere. Take a while to find it in my stuff here, but we actually done that very thing in the RAC. We've been trying to find a place to put it. It's a different dose. We actually used 1 milligram per kilogram dose of DFP, which is right on the threshold of toxicity, and we haven't done sort of a whole dose effect, and so we've got this sort of standalone stereology study. We hadn't found where we're going to put it. I don't think it's a standalone paper, so we need more to go with it, but we have the data. I could actually shoot it to you. The slide.
Speaking of dose effect, I just wanted to get a framework of your first part of your talk when you spoke of low dosages, under the threshold dosages. How would that relate to real life situations? The dosages?
Excellent point. I think it would be more related to agricultural workers, pesticide applicators, people in the, like there's all this information about the Gulf War veterans that were, I've actually spoken to some of the folks here before about how much they were sort of almost lathered in OPs, but they're in their tents, they're in these fly strips, it would be more related to that than it would be any kind of trace, like a normal environmental exposure to just somebody walking down the street. It would be well above that. We're not actually studying trace OP exposure. It's all below the level that causes acute toxicity, but certainly we haven't even studied doses in animals that are below the cholinesterase inhibition level. So like for pyrophos, you'd have to get down around 2 or something like 2 mg per kilo or something. That's excellent.
Maybe just one small question. It relates, I think, to what Fiona had said earlier. We think in medicine of pyridostigmine as being something that isn't CNS penetratable normally, at least in large amounts. But I think that a study that could investigate whether subtle changes in exon transport could be seen with PB exposure could be really exciting.
Yeah, I know. We could do it both start in culture, I guess, but then go to the animal. We probably also, I know I've read papers, I don't remember the specifics, but the idea would be that if you are in this heat and stress exposure, right, your blood-brain barrier gets compromised and it gets
as
in the
you
brain.
say, to add on some of the, or did Beatrice say that, in combination, yes.
Well, I think what Dr. Cherry might have been also trying to say is that exposures themselves, including acetylcholinesterase inhibitors that cause oxidative stress, make the blood-brain barrier leakier. Yes. So exposures, including to PB on a repeated basis, may actually facilitate entry of PB into the CNS.
Yes. But one of the things I've always wondered about the pyridostigmine story is the fact it's been used for so many years for myasthenia gravis, and you don't hear about all these other problems. And so that would make the argument that there's that additional stress, heat, oxidative in the Gulf War theater that might be the difference.
Other questions?
I just have one. You said you haven't really studied mining yet?
Have not.
Are you planning to?
Depending on money. So we haven't even proposed that yet, but yes, I definitely would like to go there.
Because you've done like the intergrade, but not the retrograde. So I think it's going to be super interesting.
We need to do it.
see both effects, right?
Yeah, absolutely.
Yeah. Thank you.
Thank you so much.
Thank you very much for having me.
It is exactly 12:45.
Schedule.
As our agenda says, we will break for lunch. Never happens.
It's a remarkable breakthrough.